Hi everyone, this is the Edexcel A-Level Biology B syllabus, and this is Paper 2. Paper 2 covers the following topics from 1 to 10. This paper is 1 hour and 45 minutes long, as you can see here, and the total marks for this paper is 90. Alright, let's get started. Alright, question 1. Carbohydrates and proteins are important biological molecules. Diagram part A. The diagram shows some of the bonds found in biological molecules. All right, let's start off with V. V is hydrogen bonding, and that is found between water molecules, for example. W, you can see the ester bond here, and ester bonds are found between fatty acids and glycerol. So remember, a triglyceride consists of one glycerol and 3 fatty acids, right? So you'd find an ester bond between them. X is the glycosidic bond. So that is a bond that we see within carbohydrates. Y is a peptide bond. So this is between proteins. And Z is a disulfide bridge. We can see the 2 sulfur atoms there. And a disulfide bridge is basically a bond between the 2 amino acids called cysteine. So this is found in the tertiary structure of proteins. All right, part 1. Which of these letters show a bond that joins glucose molecules together? The answer for that would be B. So it is X. All right, the next part. Which letters show bonds that hold together the tertiary structure of a protein? So tertiary structure is between R groups, so that would be things like the disulfide bridge between cysteine amino acids, and also hydrogen bonding as well. The other 2 that are found in tertiary structures are hydrophobic interactions and ionic bonds. So the answer for that would be B. Part 3, which letter shows a bond that holds the cellulose molecules within a cellulose microfibril? So if you had a chain of cellulose microfibrils, the interactions that hold them in place are the hydrogen bonding. So that would be V. So that's that. All right, let's move on to part B. Raw egg white is a liquid contained dissolved ovalbumin, a globular protein. It has some amino acids with polar R groups and some amino acids with non-polar R groups. If it's heated to a temperature above 70 degrees, it irreversibly changes into a solid. All right, the diagram shows how the structure of ovalbumin changes when heated. Explain why heating ovalbumin to temperatures above 70 degrees causes egg white to change from liquid to solid. So we can see denaturation happens here. I'm going to write out the paragraph. So these are the points that I've included here. Number 1, you need to mention that when it was a raw egg white, that the non-polar groups were on the inside, whereas the polar R groups were on the outside. So that would be, you can say either of these points. And because the polar groups were on the outside, that allowed for it to form bonds with water. So that's 2 marks. When we heat things up, the protein denatures. That's the 3rd mark. And then what happens is the non-polar R groups are able to form attractions with each other. And that's how you'd form the solid. All right, let's move on to question 2. Bromelain is a protease enzyme in pineapple juice. Part A, which graph shows the energy change that occur in a typical enzyme catalyzed reaction? All right, so enzymes are responsible for lowering the activation energy, and activation energy is basically, so the answer for this is W, so I'm just going to write A. But this would be what the activation energy. This would be before the enzyme is added. And then here, when the enzyme is added, you can see the activation energy has lowered. And the definition of activation energy is the minimum amount of energy required for a reaction to take place. All right, part B, a student investigated the digestion of gelatin protein by pineapple juice. This is the method used. So they created a solid disc of gelatin protein with a depth of 1 centimeter in a Petri dish, cut out a circular well with a diameter of 2 centimeters in the center of the disc, fill it with pineapple juice, incubate it for 1 hour at 37 degrees, and then measure the diameter after it's been digested. All right, the pineapple juice, the diagram shows the gelatin disc before and after incubation with the pineapple juice. And then the next part, it says the diameter of the circular area that had just been digested after 1 hour was 6 centimeters. Calculate the volume of gelatin that has been digested. Okay, so we're given the formula of the volume of a cylinder. Okay, so we need to figure out the volume before and after. So, okay, let me write this down here. This is the before, this is the after. So I'm just going to write before, after. Now the depth is going to be the same. It is both 1 centimeter. Whereas the radius before, the radius was... Okay, the diameter was 2 centimeters, which means the radius was 1 centimeter originally. So that means before would be r, or pi, 1 to the power of 2 times 1. And then after would be pi times, okay, the final diameter was 6 centimeters, which means the radius was 3. So 3 squared times 1 as well. All right, let's calculate that. Okay, this was 3.14 and this is 28.26. And then we just have to do 28.26 minus 3.14. That is the difference. And that gives us 25.12. Now all of this is in centimeter cube. We need to convert this to millimeter cube, so we have to times that by 1,000 because that's what the answer is expecting of us. So that would be 25120 millimeters cube. All right. Oh, before I forget that, it's written in 2 significant figures. So that would be 25000. All right, let's move on to part C. Bromelain also digests other proteins. The student also investigated the effects of changing the concentration of a soluble protein and the addition of copper sulfate on the rate of digestion of this soluble protein by bromelain. All right, part 1, it says, explain the effect of increasing substrate concentration on the rate of digestion of the soluble protein. So this is what I've written. First, you would note down the trend, right? So as the concentration of substrate increases, the rate of digestion increases. And then I wrote at a higher concentration, the rate of digestion plateaus. The second thing you need to mention is the reason why the rate of digestion is increasing is because there's a greater chance of the substrate and enzyme colliding successfully with each other. And then lastly, the third mark is it plateaus because the active site becomes occupied. All right, let's move on to part 2. Explain the effect of copper sulfate on the digestion of the soluble protein. So we can see from this graph that if you keep adding copper sulfate, it'll lower the maximum rate of digestion. And the only kind of inhibitor that does this is a non-competitive inhibitor. If it was a competitive inhibitor, it would look something like this, in which eventually it would reach the top. But in this case, it's not, so it's non-competitive. All right, so this is what I've included. Copper sulfate is a non-competitive inhibitor. That's the first mark. How does it work? It binds to the allosteric site and changes the shape of the active site. And then the third mark is therefore the substrate can no longer bind. All right, let's move on to question 3. The photograph shows part of a testes as seen using a light microscope. Part A, it says the cell labeled X is a primary spermatocyte. At which stage of cell division is cell X? All right, so I've included a diagram here to show you spermatogenesis. At first, the spermatogonium undergo mitosis, and then it starts to form the primary spermatocyte. So that's when it starts to undergo meiosis 1, and then when it forms your secondary spermatocyte, that's when it's undergoing meiosis 2. So the answer for this would be A, prophase 1 of meiosis. All right, part 2. In the photograph, cell X has a width of 0.8 centimeters. The magnification of the photograph is 1,500. All right, the equation for this is magnification equals image size over actual. In this case, the image size is, we need to rearrange the equation so that the actual is on one side. So that would be I over M. Now, because it wants you to answer it in micrometers, I'm going to convert this 0.8 centimeters to micrometers first by timesing it by 10,000. 1 millimeter equals 1,000 micrometers, and we know that 1 centimeter equals 10 millimeter. That's why I'm timesing it by 10,000. So 0.8 times 10,000 gives us 8,000. Now we can plug it into this expression here. So 8,000 divided by 1,500, that gives us 5.33, but it says give your answer to one decimal place, so it'll be 5.3. All right, part B. Some pregnancies do not develop to full term, so no baby is born. Damage to the DNA of sperm is thought to be one of the causes of pregnancies not developing to full term. Scientists investigated if there is an association between production of sperm with damaged DNA and the risk of pregnancies not developing to full term. The percentage of sperm cells with DNA damage was determined for 130 men, where there was history of pregnancies not developing to full term. The percentage of sperm cells with DNA damage was measured for 78 men. Let me just highlight that, where pregnancies did develop to full term. So this is full term, this is not to full term. All right, so these are the results. Let's look at part 1. Calculate the number of men with partners that have a history of pregnancies not developing to full term that have 25% or less sperm with damaged DNA. Okay, so for this, we have to scroll up to the first table. So it's asking for 25% or less, which means we have to add up these 4 bars here. So that is 2 plus 4 plus 3 plus 3. That gives us 12. All right, we're going to write 12. The scientists concluded that having more than 25% of sperm with damaged DNA leads to a high risk of pregnancies not developing to full term. Analyze the data to evaluate this conclusion. So whenever it comes to this kind of question, we have to look at the evidence that support this statement and evidence that goes against. So let's say if we look at this graph, the first one shows you that most men have more than 25% of sperm cells with damaged DNA that do not have pregnancies coming to full term. And if you look at the second graph, we can see that most men that do have pregnancies coming to full term have less than 5% of sperm cells with damaged DNA, which is what I included here. So the majority of men that did not have pregnancies come to full term have more than 25% with damaged DNA. That's the first mark. The second mark, I included this here. Most men that have pregnancies that come to full term only have 0 to 5%. So that's the second point. So some statements that can go against is, you can see that even though these people had less than 25% of sperm cells with DNA damage, their pregnancies or their partner's pregnancies did not go to full term. So I wrote that here. Although there were some men that did not have this one here. All right. Now, another thing we can always comment on is the study itself. So I mentioned that it's a small sample size. Small sample size because there's only 208 men. And there might be other things that might have contributed to these pregnancies not coming to full term. So maybe things like their lifestyle or their diet or their age, and that might have played a role. So this is the third mark, this is the fourth mark, and this is the fifth mark. All right. Let's go on to question 4. Salt marshes are coastal habitats where silt is deposited. Salt marshes are colonized by different species of plants and are regularly flooded with seawater. All right, part A, the conditions close to the sea are very windy. Part 1, which row of the table causes the highest rate of transpiration from plants? When it comes to transpiration, it involves water vapor in the air spaces of the leaf, leaving the leaf via the stomata. So I'm just going to draw a leaf so you guys can, that's a terrible leaf. So you have water vapor here, and it leaves the leaf via the stomata, via diffusion. All right, so in terms of temperature, we know that the greater the temperature, the higher the kinetic energy, which means the particles are moving more quickly, which means the rate of diffusion is greater. So that means it's going to be high. Now with wind speed, if you have a lot of wind, that causes it to blow onto the water vapor surrounding the leaf, and that would cause a steep concentration gradient, meaning that this would be inside of the leaf. So inside of the leaf, you'd have a lot of water vapor, and then outside of the leaf, you would have very little water vapor. So the steeper the concentration gradient, the faster the rate of diffusion. So this would be this one here. Lastly, air humidity. Air humidity refers to the amount of water vapor in the atmosphere. So again, we want to have a steep concentration gradient in this case, and you need to have low humidity for that for the rate of diffusion to be quick. So the answer for that is B. Part 3. Samphire is one of the few plants that can grow in the areas close to the sea. Samphire plants have very small leaves and swollen stems. The diagrams show a samphire plant and a cross-section through the stem of the samphire plant. Explain how it's adapted to grow in areas of salt marshes closest to the sea. So just to recap, it is places that's regularly flooded with sea water and very windy. That's something to keep in mind. All right, so here's what I wrote. I wrote that the cortex containing cells with a high salt concentration ensure that there is a lower water potential inside of the leaf, therefore less water loss via osmosis. I also mentioned the thick epidermis. You can see that here. So thick epidermis and reduced surface area of the leaf, the fact that it's like this and not like a regular large leaf, would also reduce water loss via transpiration. So that's third and the fourth mark. Some points you can also say is talking about the air spaces which allow for gas exchange, so oxygen for respiration, carbon dioxide for photosynthesis. You can also talk about how it has a lot of chloroplast for photosynthesis. And so the stem can undergo photosynthesis. All right, part B. A group of students investigated succession in a salt marsh. The students measured factors at 5-meter intervals along a transect starting at the edge of the sea. The factors that were measured were ACFOR, scales for these three things, index of diversity for all plant species, and percentage of silt made up of organic material. All right, so the ACFOR scale is a scale that measures or compares the amount of species in a given area. So A is for abundant, C is for common, F is for frequent, O is for occasional, R is for rare. And sometimes you might have none as well. All right, so part 1, it says give 2 limitations of using the ACFOR scale to compare the distribution. Well, first of all, it's a category, it's not a quantitative scale. And because it's not a quantitative scale, you can't perform statistical tests on them. So you don't know if there is a significant difference between the categories or the places that you are surveying. Another thing you can also mention is that it's quite subjective as well. All right, part 2. Salt marshes are produced by deposits of silt from rivers. As the distance of the sea increases, the age of the salt marsh increases. Explain the changes in distribution of the species as distance from the sea increases. All right, so again, this study is about succession, which is when a biological community changes over time. Now, they mentioned here that as the distance of the sea increases, the age of the salt marsh increases, which means these would be the older salt marsh and these would be the younger ones or recent ones. And so the opposite would be the same. So the closer the area is towards the sea, the younger the salt marsh would be. Now, the species that are thriving closest to the sea are the samphire, right? You can see the sea lavender and the scurvy grass are absent. This would mean that the samphire, they are the pioneer species. So a pioneer species are species that can colonize a newly exposed land or exposed environment. In this case, we have the deposits of silt coming from the river, and that would create new land for them to colonize. So the samphire, they're able to stabilize deposits of silt from the river, and then when they died, you have the microorganisms that were able to decompose them, and when they decompose them, they released nutrients into the soil. When there's more nutrients in the soil, that means that other plant species can start to grow. Now, like I mentioned earlier, the further it is away from the sea, the older the salt marsh is. Now, we can see that the species, the index of diversity for all plant species increases. And we can also see that the samphire becomes absent, and you've got more sea lavender and scurvy grass away from the sea. So I wrote that here. So we can see as the distance from the sea increases, the age of the salt marsh increases, the plant diversity increases as well, although that's not really a mark here. Plants like scurvy grass took over, which meant that succession takes place. And again, succession is when there is a change in the biological community over time. Now, when it's further away, we saw that the samphire is absent. So you can see that there's a change in the community there, right? There are less samphire away from the sea. I should say there is no samphire away. So there is no samphire away from the sea due to competition with the scurvy grass and sea lavender. So we have 1 mark for pioneer species. Second mark is succession took place. Third mark is being able to stabilize the soil. Fourth mark is increasing nutrients when they die. And fourth mark would be competition, right? So that's why there's no samphire away from the sea. Okay, let's move on to question 5. The human retina contains photoreceptors called rod cells and cone cells. Part A1, which of the following occur in the rod cell when it is stimulated by light? Alright, so I've included 2 diagrams here. This is just to show you what the rod cells and the cone cells look like. We'll get to this image later. But you can see that rod cells here are connected to these neurons called bipolar neurons. Now let's scroll down to this diagram. So the way the rod cells and the bipolar neuron work is a little weird. So when it's dark, the sodium ions get pumped out of the rod cells via active transport. This creates a concentration gradient, which causes the sodium ions to diffuse back, and you can see it there in that diagram. When sodium ions come in, because sodium ions are positively charged, that makes the rod cell more depolarized. You can see that there, becomes more depolarized. When it is depolarized, the rod cell releases neurotransmitters, which are able to diffuse to across the synapse to the bipolar neuron. Now, what's interesting is that these neurotransmitters are inhibitory. So if it's inhibitory, it prevents an action potential from being generated at the bipolar neuron. However, when there is light present, something else happens. In the rod cells, you have these light-detecting pigments called rhodopsin. So when light is absorbed by it, it gets broken down to something called retinal and opsin. Now, when this happens, the cis-retinal changes shape to transretinal. And what that does is ultimately block the sodium ion channels. So you can see previously sodium ion channels were allowed to diffuse in, but because of this change, now the sodium ion channels get blocked, right? And that prevents sodium ions from going into the rod cells. Now that causes, because you have the sodium ions that are being actively pumped out, that eventually causes the rod cells to become more and more negative, right? Until it becomes hyperpolarized. So hyperpolarized just means that it's more negative than its resting membrane potential, which is minus 70 millivolts. So because it's hyperpolarized, then that prevents the release of the inhibitory neurotransmitter. And because that doesn't get released anymore, then an action potential gets generated at the bipolar neuron. So to answer this question, what happens when the rod cells are stimulated by light? The first statement is correct. It gets broken down to these 2 things. Number 2, there is a decrease in the release of the neurotransmitters. So these 2 statements are correct, because remember, the rod cells become hyperpolarized. And lastly, the third statement is incorrect, because if you recall, the sodium ion channels get blocked, and that prevents the diffusion of sodium ions into the rod cell. So the answer is A. Alright, I've just kept this diagram here, but let's go over parts 2. Explain why the center of the retina is less sensitive to low intensity light than the outer areas. Alright, so when it says the center of the retina, it's referring to the fovea. So the fovea is any light that lands on our fovea creates the sharpest image. So the fovea is where you'd have the highest density of cone cells. So remember, rod cells are good for detecting low light intensity. Cone cells are very good at detecting color. So let's get back to this diagram. There's this term called spatial summation. So what I mean by that is Here we have multiple rod cells that are connected to one bipolar neuron. Whereas for cone cells, you have one cone cell connected to one bipolar neuron. Now, spatial summation is basically when you need multiple neurons or multiple receptors in order to affect the membrane potential of the neuron. In this case, you need multiple rod cells that would converge to a single bipolar neuron. That's why rod cells are a lot more sensitive. Whereas cone cells, you need brighter light, greater stimuli in order to trigger an action potential in the bipolar neuron. Let me write this down. All right, this is what I've written down. Let's go over the marking scheme. So the first mark is more cones, and I wrote the fovea has more cone cells. The second mark is cone cells are less sensitive to light. And then lastly, the third mark is because one cone cell is attached to one bipolar neuron. And you can even mention the word spatial summation. That will give you the same mark though. So those are the 3 points. All right, let's go over part B. Red-green color blindness is a sex-linked genetic condition. People with this condition are unable to detect differences between red and green colors. The pedigree diagram shows the inheritance of red-green color blindness in a family. The allele for red-green color blindness is recessive, and the one for colored vision is the dominant allele. All right, part 1. How many individuals in the family must have a genotype of this? So that's heterozygous. All right, so if I've just zoomed in a little bit, so an individual that is heterozygous has to be female, and they do not have the color blindness. I'm gonna shorten this to color blindness. All right, so great way to work this out is to do it backwards. So you start with the recent generation. So here, let's say individual 8 is female. So if it's circle, it means female. And this person has color blindness. Therefore, they are homozygous recessive. Now, if they're homozygous recessive, one allele must come from the father, the other allele must come from the mother. So that means this mother must be heterozygous. So individual 4. I'm just going to write the people first. So persons, 4. Now let's look over to the right hand side. We have individual 11. Individual 11 is a male, so that means it's XY. And this person has color blindness, so there has to be an R there. So like I said, each chromosome must come from each parent. The Y chromosome must come from the father, which means the X chromosome came from the mother. And so this person must also be heterozygous for that allele. Ooh, another individual that I'm missing is number 10. So for example, individual 3 is a male, right? Now individual 3 does have color blindness, that's recessive. Now, for this female, which is individual 10, they must inherit the X chromosome from the dad. And that's the recessive one. And must inherit the dominant allele from the mom. So actually, individual 10 is also heterozygous. All right, the last individual is person number 2. Right, the reason why is individual number 1 is a male, so I'm going to write XY. But this person does not have color blindness, meaning they have the dominant allele. Whoops. Okay. Now, they have 2 kids, number 4 and number 6, that are heterozygous. Now, if they're let's take number 6, for example. If they're heterozygous, it means that they take the dominant allele, or the one with X capital R, from the dad. That means the lowercase r must come from the mom, and the mom does not have color blindness, so therefore she is a carrier. So these are the 4 people that have that are heterozygous. So the answer is C. I'm going to zoom out now. Explain how this pedigree diagram demonstrates that red-green color blindness is caused by a recessive allele. So there are multiple ways to answer this question. This is how I chose to answer it. Person 6 and 7 are not colorblind, but they do have a child with color blindness, and that means the parents must be carriers for that allele. And you can see that there, 6 and 7 don't have color blindness, but 11 does. Now, there are other examples that you can write here. So I'm just going to read out the marking scheme. You can talk about how with individuals 3 and 4, it produces a kid with color blindness. That means that individual 8 inherited the recessive allele from both parents. You can even talk about how 1 and 2 produces 4 and 6, which are carriers for the recessive allele. But I decided to go for this family instead, just because if you have 2 parents that don't have the condition, but a child that does, that indicates that it is a recessive allele. So that's the more obvious one, at least for me. All right, part C. Pingaleb is an isolated island in the Pacific Ocean. I hope I'm not pronouncing that wrong. I probably am. On Pingaleb, between 4 and 10% of the population has a condition called achromatopsia. This means that they're unable to see any color. In most other countries, the incidence of achromatopsia is around 0.003%. The pedigree diagram shows the people who are and who are not affected. Deduce how achromatopsia is inherited. Use information in the pedigree diagram to support your answer. Okay. All right, so in order for you to get the 3 marks, you need to mention whether or not it's caused by a dominant or recessive allele. And then you also have to mention whether or not it's a sex-linked disease or a sex-linked condition. So just like with the previous question that we saw, if you see 2 parents that do not have the condition, but a child that does, then that means that the condition must be caused by a recessive allele. And that's what I've included in this text that you see here. It's caused by a recessive allele. That's the first mark, because individuals 4 and 5 do not have the condition, but person 6 does. So it's this whole sentence here. That would be the second mark. Now, you also need to mention whether or not it is sex-linked. In this case, it is not a sex-linked condition because person 3 was not affected. Let me explain. Okay, so let me assign some letters. Let's say XA is a normal allele, X lowercase a is achromatopsia. So individual 2 is female, and she must be homozygous recessive for it. Now, this person, individual 1, does not have the condition, so he has the dominant allele. Now, for person number 3, they inherited the Y, because person number 3 is a guy. They inherited the Y chromosome from individual 1, which means they must have inherited it from, if it was sex-linked, they must have inherited the recessive allele from their mother. And in this case, individual 3 is not affected. That's why it is not a sex-linked disease. I'm just going to erase everything. Okay, let's move on to part 2. In 1755, a tsunami reduced the population to 20 people. There has been little immigration to the island. In 2022, the human population of Pingaleb was approximately 250. So that is a drastic decrease in population. Explain why the incidence of achromatopsia is higher in Pingaleb than in other countries. All right, let's go over what I wrote. So this is because there is a genetic bottleneck. So you do need to mention this term. A genetic bottleneck is when you have a drastic decrease in population and all of a sudden the number of available alleles that you have has decreased, which is what I mentioned there. Reduced gene pool. That is the second mark. And then therefore the chances of 2 carriers mating to produce a child with achromatopsia is higher. That is the third mark. Another thing you can also say is this population is isolated. So there are few new alleles that get introduced into that population. That's another point that you can also say. All right, so we've gone over questions 1 and 5 for this paper. If you want to check out the next few questions, you can look at part 2 of this video. Thank you, and if you have any questions or comments, please write them in the comment section below. All right, bye and good luck.